Hello, everyone, and welcome to this session of ACS Science Talks, Connecting the World Through Science. This is the virtual lecture series, Scientific Talks by Specialists on Specialized Topics for a Specialized Audience. I'm Neha, and I'm pleased to be your host for today's broadcast of ACS Science Talks. I'm joined by my colleagues Dr. Sriranjani Pulakkat and Dr. Ashish Mahto, who will be moderating today's live Q&A. In its efforts, ACS provides a variety of resources. One of the flagship ACS resource is the ACS Meetings. Just like ACS Science Talks is a platform by ACS to connect researchers, ACS Meetings aims at unifying the scientific community. Every year, active researchers and professionals from across the globe come together to share ideas and advance the scientific and technical knowledge. ACS meetings are regularly attended by thousands of science professionals every year. As we mentioned about the goals and mission of ACS to support the global scientific enterprise, ACS has taken the challenge of the COVID-19 pandemic and converted it to an opportunity. ACS meetings have embraced the hybrid model, which is helping us to enhance the networking opportunities by combining in person interactions with virtual networking platforms. It promotes inclusion and accessibility by removing geographical barriers. It enhances representation by reaching out to diverse range of presenters and attendees. It provides flexibility and convenience by accommodating different preferences and circumstances. It also increases engagement and interaction to foster a dynamic and inclusive conference environment. It provides continued knowledge sharing opportunities by ensuring continued of knowledge during unforeseen circumstances. And lastly, it promotes sustainability and cost effectiveness by eliminating the need for travel and accommodation expenses and the associated carbon footprint. Our speaker for today is Dr. Wenwan Zhong. She is a professor of chemistry at the University Science and Technology of China. Dr. Zhong earned her BS degree from USTC and her PhD degree from the Iowa State University. After working at Los Alamos National Laboratory as a postdoc, she started her independent academic career in the University of California, Riverside in 2006 and became a full professor in 2016. She also served as the director of the Environmental Toxicology Graduate Program at UCR from 2018 to 2023. She received the Career Award from National Science Foundation in 2011 and the UCR Chancellor's Award for Excellence in Undergraduate Research in 2019. She is also a topic editor of the ACS journal Precision Chemistry. Dr. Zong's research is devoted to developing innovative analytical techniques to advance our understanding on how biomolecules function, to improve disease diagnosis and treatment, and to assess the potential impacts from exposure to environmental pollutants. Her group employs synthetic receptors, functional nucleic acids, and nanomaterials for target recognition and signal amplification, and also uses open channel separation such as capillary electrophoresis and flow field flow fractionation, optical spectroscopy, and mass spectrometry to assist with marker discovery and analysis. Thank you so much, Dr. Zong, for joining us here today. The stage is all yours now.
Thank you, Neha, for the introduction. So I'm going to share my slides. Can you see my slides fine?
Yes, yes.
Okay, great. So it's my great pleasure to be able to speak here and discuss some of the results we obtained from our work. So currently, as Neha introduced, I'm a professor of chemistry working in the University of Science Technology of China. So I moved to this university actually last year, establishing a new lab here. Before that, between 2006 and 2023, I actually worked in the Department of Chemistry in University of California, Riverside. So All of the results I will present today actually were obtained at UCR. So hopefully in the near future, I will be able to present to you some of the new results my group actually obtained in this new location. So my topic is the biosensing with arrayed deep cavitants. However, I also would like to take this chance to introduce all the works actually briefly going on in my briefly introduce the work going on in my lab. So as you have heard from the introduction, my lab actually is basically an bioanalytical chemistry lab. So our focus actually on biomarker discovery and detection. So aiming to improve disease diagnosis and also to assist therapeutic implementation. So we actually use a lot of separation tools, including open channel separations like flow fractionation and capillary electrophoresis, as well as microfluidic devices to obtain to purify molecules and do a lot like a large scale profiling, so to assist with biomarker discovery. And then for detection, we actually employed chemical tools, including nanomaterials, functional nucleic acids and synthetic receptor, etc., to help us improve the signal to achieve high selectivity, and of course to simplify the detection implementation. So using with these two goals in mind, we actually mainly work on 4 areas. The first one is actually to do molecular profiling on single bioparticles. So mainly using fluorescence detection or imaging, we try to analyze or colocalize multiple cargo molecules on the single extracellular vesicles. And extracellular vesicles actually are very promising biomarkers. People have used them in liquid biopsy for disease diagnosis and also for monitoring therapeutic effects. However, they are highly heterogeneous. So they have diverse cargoes enclosed, and then they could have many different kinds of vesicles exist simultaneously in biological samples. Therefore, we try to use the strategy of single particle analysis to overcome the heterogeneity in EV analysis. So mainly we actually amplify the signal on each particular proteins or nucleic acids carried by one single particle and enlarge the size by using nanomaterials like DNA nanostructures. And then we can count them individually based on the different signals, fluorescent signals emitted from different markers. Using that, we could actually differentiate the early stage cancer patients from healthy people. Also, we are interested in analyzing the bicorona formation, which is part of the nanobio interface field. It's a very interesting topic. So mainly when nanomaterial go into biological matrix, they could absorb, because of their high surface energy, they could absorb a lot of biomolecules. And this type of biological coating, which we call it biocorona, actually will mask their original material. So then when they interact with cells, they're not directly interacted from their own surface, they actually interact through by the bridge formed by this biocorona. Therefore, we would like to use all these analytical tools available to us to improve our understanding of how this biocorona is formed and how the formation is actually governed by the properties of the nanomaterials, so that we could understand better how the biocorona could contribute to the biological significance carried by the nanomaterials entering biological systems. Then once we kind of discover very meaningful biomarkers, such as the extracellular vesicle subpopulations highly related to disease, we then develop many methods to improve their detection. And hopefully we can detect them at very low concentration and with strong signals. And in addition to extracellular vesicle, we are also interested in other kind of markers present in biospecimens. For example, circulating DNA and small RNAs, as well as the proteins. So for these biomarkers, our goal is to amplify the signals using different strategies, in particular isothermal nucleic acid amplification. And then we also want to improve their purity by purifying them and enriching them from the complex matrix. Then we can clean up the background and obtain very clear signals. Then the last areas, but not the least, of course, is what we will focus on today, which is to exploit the host gas interactions for differentiation of biomolecules. So this kind of sensing strategy is very different than what we use in the liquid biopsy approach. In liquid biopsy approach, we actually employ antibodies or aptamers, this kind of very specific probes to highly selectively recognize our markers, our targets. However, in the concept of like host gas-based sensing, since the hosts usually have pan-selective binding to a variety of molecules that carry similar chemical part of their structure could be quite similar, but then they may lack of selectivity if they are used individually. Then our strategy actually is to mimic how our nose works. So we exploit a bunch of receptors and combine them with several gas, and then forming the host gas sensors. And then when we use several sensors all together, we actually can get the pattern recognition works on to specifically recognize a target. Then I will go through more details in my talk today. So for this time for sensing, we exploit host gas interaction. Then let's take a look at how these synthetic receptors actually bind with their gas. So there were actually many factors that could influence the binding strengths as well as specificity between receptors and their gas, even though they could have certain selectivity. Then in particular, they actually rely on complementary molecular recognition. So if the gas have matching shape or matching size with the host, with the binding cavity of the host, then they will allow this kind of like a lock-in key binding model or infused fit binding model to work. Then they can gain high binding strength and also high specificity. Then also the binding can be strengthened by many different types of non-covalent molecular interactions. For example, electrostatic interactions, hydrogen bonding, and also the pi bond-based interactions like a cation bond or pi-pi stacking. And of course, between molecules, molecular weight could play an important role in enhancing the strength of interactions. But therefore, the van der Waals interaction also play a role in enhancing the host gas interactions. In addition to this molecular interaction, depending on what kind of solvent they are in, the solvent will play a role as well. For example, for what we are interested in, like sensing biomolecules, all our systems work in aqueous solutions. Therefore, the hydrophobic effects would be highly influential to the binding strengths, and because the cavity usually could be hydrophobic, and then the hydrophobic interaction will drive the hydrophobic portion of the gas molecule to go into the cavity. Then, of course, thermodynamics always are important things. So then they could, for example, entropy can contribute significantly to the binding strengths. For example, in aqueous solution, they have a lot of hydrated water molecules, and then these water molecules could leave the host once the gas bind to the host, and then the leaving of water molecule increase the entropy, so then they can enhance the free energy change. Then compared to protein-based binding, these synthetic receptors actually have certain advantages. So first, they could be produced in a large scale at a relatively lower cost. And then the structures can be designed to tailored for maximizing binding selectivity. And also they could be very flexible in applications. For example, they are relatively small compared to antibody. So then we have used them in open channel separation, or they can be immobilized on columns to enhance target recognition. And also we have used them in diverse to form optical arrays, and optical detection are very simple, and then they are very versatile. So we have applied them to detect a large variety of biomolecules. So here I show the structure of the deep cavitants that we used, which are the workhorse in our sensing array. So these are water-soluble synthetic receptors that macrocycles. They have aromatic structures, actually the scaffold, aromatic scaffold, and then forming the hydrophobic cavity. Then their rim actually could fold it could self-fold, and then also strengthened by hydrogen bonding, then forming this waist-like shape, and then they can bind to the size matching gas to their cavity. And then their upper rim and also the lower rim actually can be tailored. So then by changing the structure of the upper and lower rims, they actually can influence the selectivity and then affect the structural rigidity as well as their solubility. So all of these chemicals, including both the host and the guest, are synthesized in my collaborator, Dr. Richard Hooley's lab at UC Riverside. We have worked together on these topics actually for ever since 2015. So hopefully we will be able to continue the collaboration, this fruitful collaboration. So for a cavitant to bind to their guest, first of all, if the gas is neutral, they can bind via the hydrophobic effect into their cavity. So when in this case, then hydrophilicity of the gas increase actually will decrease the binding strength. So therefore, you will see decrease in their affinity constant, binding affinity constant. But then also the trimethyl ammonium head actually can have very strong affinity, very good affinity to this kind of deep cavitant. For example, acetylcholine actually displayed very nice binding affinity in water to deep cavitant, although this molecule is actually water soluble and hydrophilic. This is to take advantage of the cation pi interaction between the aromatic structure within the cavity as well as the cationic head on the trimethyl ammonium. Then we actually can modify the rim, like I mentioned before, so then and also have different lower rim structures to increase the diversity of our host. Then we could use an array of synthetic receptors as biosensors. So in biosensing using synthetic receptors, there is typically the strategy of indicator displacement. So that means if we have a fluorescent molecule, like a dye molecule, originally it can emit strong fluorescence, but when it binds to the host, its fluorescence could be quenched. So this is only one example. So we also could see it's a fluorogenic dye that once it binds to the host, its fluorescence could be enhanced. But here we just use the fluorescent dye as an example. And then in this example, we assume that when the dye binds to the host, its fluorescence quenched. Then once the target actually is in the system, if we have the target molecule in the system and the target actually can have a high affinity to the host compared to the dye itself, then we could see displacement of the dye from the host. Then this kind of displacement could turn back on the fluorescence of the dye molecule, since now it's being coming off from the host. Then taking advantage of this kind of indicator displacement assay, we can also arrange a bunch of host and guest elements and then arrange them in the array format. Then even without very high specificity recognizing a very particular target, then we can actually use an array of dye and host combinations to produce optical signals. The pattern of the signals actually can be interpreted using statistical tools. Then we can do differentiation and classification of the molecules. In actually using this type of differential sensing approach, we find that we could actually differentiate very small differences between biomolecules. For example, the chemical modification on peptides, as well as the topology of the folded nucleic acids, the topology differences of the folded nucleic acids. Along the way, we also find that in addition to the host-gas interaction, we could actually add more variables to our array to improve its performance. For example, we could add additives to tune the host-gas interaction. So with additional molecules in the system, we can actually build a larger array to reveal more tiny differences which may not be able to do by just host and gas interaction. We also could have an indirect sensing strategy. Actually, it's not just rely on simply host and gas interaction. We could have our target play a role in the recognition as well, in the signal change as well. So then the tiny actual differences in the target could actually could bring in change in the signal, so then we can do better differentiation. So I will talk about all these different things I would pick a few examples from our previous work to demonstrate all four of these concepts. Then, like I mentioned, we use these arrays to sense small changes on macrobiomolecules, and those mainly include a non-canonical folding in nucleic acids, and also the chemical modification on peptides. For example, methylation on lysine, and also phosphorylation on serine and other amino acids, and also peptide isomerization. So today, I actually mainly will focus on how we design the arrays and apply them to detect lysine methylation, as well as phosphorylation on peptides. And then also I will talk about work on how to differentiate non-canonical foldings, different types of non-canonical foldings in nucleic acids. So first, in the analysis of peptide post-translational modification, actually this we actually take advantage just as the indicator displacement format. So in this case, we actually exploit the binding between the cavitand host and then the trimethylammonium head group, and then this interaction could be interrupted by the methylation on lysine. Because the lysine itself actually is a trimethylammonium, and then if the trimethylated lysine actually is one type of this kind of very good guest for cavitans, then if we have the fluorophore and we put the trimethylammonium binding handles on this fluorophore, then we can introduce binding between the host and this fluorophore. And then the binding could induce fluorescence change. Usually it quenched the original fluorescence of the fluorophore. And then when we have the methylated peptide in place, then we can displace this off and then turn the fluorescence back on. Like I said, this could also be reversed, working in the reverse way. So the gas can be turned on when bind to the receptor, and then once displaced, the fluorescence can be turned off. So anyway, we need to introduce the fluorescence change in this type of sensing strategy. So at the beginning, we focus more on the histone methylation, just because for this post-translational modification is a very important epigenetic variation. So in addition, so this is work outside of the sequence of nucleic acid, but control protein expression by changing the interaction between histone and the DNA. So when we have, for this methylation, actually there were many different kinds of situations. So like for the lysine could locate at different location on the protein, and then the methylation could be monomethylated, dimethylated, or trimethylated. So this kind of the site selectivity as well as the state selectivity of the methylation is very important actually for the function of the methylation to regulate the gene expression. And therefore, they are tightly controlled by this by variety of enzymes, including writers, which put the methyl group on, and the erasers, which is to remove the methyl group. Therefore, it's very essential to study the enzyme activity and then to find out new enzymes to regulate histone methylation or develop drugs to control the activity of the enzymes. All of this require like a quick monitoring of the enzyme activity. Therefore, and also the enzymes, since they have high specificity to the substrate sequence, and then we need actually state and site selectivity in our methylation recognition methods. So first development actually, since we use indicator displacement approach, the first thing to do is to obtain dye molecules that have the binding handle. So our collaborator, Dr. Huli, actually asked his group to synthesize 404 that from fluorescein, so derived from fluorescein molecule. And then this fluorescein molecule actually is coupled to a trimethylammonium head, then they achieve good affinity to the cavitin. However, if we have like only the dimethyl ammonium or monomethyl ammonium, we could see the fluorescence change once this fluorescent gas that bind to the cavitin actually would be smaller than the trimethyl ammonium modified fluorescein. So later we actually studied the quenching mechanism. We found out actually the quenching is due to the triggered anionic cavitin aggregation that bring the fluorescent molecules close to each other, so then they actually have the self-quenching effect going on, and then to cause the fluorescence decrease. Then this actually decreased fluorescence could be reversed if we have the trimethylated peptide in place. So H3 meaning is the H3 protein on the histone, and then K9 meaning is the number 9 position of the lysine on the peptide, on this particular H3 peptide that we used. And then it has the black curve showing the peptide actually carry trimethylation. And then it has the high fluorescence enhancement with the increasing with the peptide concentration. Of course, at the higher peptide concentration, it could reach a plateau. And then if the methylation state actually, the methylation level goes down, then we could see lower change in the fluorescence. And if it's just irrelevant peptides, for example, the digestive peptides from human serum albumin, then we did not see any change of the fluorescence. Using this kind of change, we actually can do like enzyme activity monitoring. So here actually we could see like the trimethylammonium level actually increase with a longer reaction time for the methyltransferase. But then if it's demethylase, then we could see fluorescence keep decreasing. And then also, since we can monitor the increase using the increase or decrease of fluorescence to represent the enzyme activity, then we can, of course, to monitor how the inhibitors could work actually to stop to like inhibit the enzyme activity and to slow down the fluorescence change. Therefore, just use one sensor if we have a relatively pure system, for example, to monitor a known enzyme's activity, then we have a known peptide. So then we can just use one sensor and just use the fluorescence intensity to tell us the activity of the enzyme. Then we thinking whether we could work on other like more diverse targets. Then we need to introduce more guests. Then our collaborators synthesized another type of guest that derived from rhodamine B. And then the rhodamin B, again, we have a trimethylammonium. The rhodamin B derivative, again, have the trimethylammonium head, so then it can bind to the cavitin. However, this kind of binding, again, although it's causing the same type of fluorescence decrease, for example, you see increasing cavitin will decrease the fluorescence of this fluorophore for this rhodamin. And then, however, it's no longer due to aggregation. Since if we put in surfactant or liposome in the system, it does not reverse the fluorescence just like in the other case. And in this case, then it would be the molecular, is the energy transfer between these two molecules and then the effect that caused the quenching of the rhodamin molecule. Then similarly, as the fluorescein, if we only use one sensor from the fluorescence change, we could see the how the methyl transfer rates work and also how the demethylates work. But still, we have quite a problem here. If only one pair of the host and guest, then it would be difficult to confirm the target identity. Like I said, if in an enzyme solution, you only have one target and one enzyme, this type of single sensor will work perfectly. And then it's very simple, we just monitor fluorescence intensity. However, if we have multiple peptides in the solution, in the mixture, but then we would like to see which peptide actually would be reacted, then we will have an issue. Therefore, we actually use various sensor elements to form an array. Then here we actually implemented different kind of manipulations, including pH condition, and as well as we have different 3 types of cavitin used in this array. And then we form a kind of relatively big one. We have 3 pH, we have 3 receptors, as well as 2 types of fluorescent gas. So then we can have random combination of all these factors. Then what we do is we can obtain fluorescence patterns for each peptide, and this peptide have the methylation, like different level of methylation, and also could have the methylation at different position of the lysine on the same peptide, or they could be methylations on different peptides. Then we use the pattern recognition tools, for example, principal component analysis to interpret the fluorescence patterns. And then we can actually project all the different samples onto a 2-dimensional or 3-dimensional space. And this is for PCA, then the results are called the scatter plot. And then when we plot the first two principal components onto this 2D space, we can see all these targets actually can be differentiated nicely. So for example, for this subset, we can differentiate different methylation states from non-methylation, mono, di, and trimethylation on the same position. And here in the middle subset, actually, we can see the trimethylation on different lysines. They are being well differentiated. And then the differentiation effect actually can be illustrated by the 95% ellipse surrounding the repeated samples. And if the ellipse is not overlapping, then we'll consider as like a good differentiation. And here, the last subset actually showing the trimethylation on not only different peptides, but also the peptides carrying other modification. It seems they can be differentiated as well. So using this kind of array, then we could actually monitor the reaction on multiple peptides simultaneously. So here we demonstrated if we have start with a mixture of peptides, but only one of these peptides could be the substrate of the enzymes we are studying. For example, this is a demethylase enzyme, and it's a K9 demethylase, so that means it only work on the trimethylation on the number 9 lysine on this peptide. So if the peptide actually has the methylation on the 27 position or the 4 position, then it will not work. So we monitor the fluorescence pattern change over time of this enzyme reaction. Then we could see So if from time 0 to after reaction of 1 hour, we could see this the fluorescence pattern of the reaction mixture actually getting closer and closer to the standard mixture that containing the demethylated methylation on K9. So, however, it will never reach this position just because this enzyme actually only remove 2 of the methyl group on this K9 site. So the final product is actually the K9ME1. So then when we have a mixture of the K9ME1, then this mixture actually overlapped perfectly with the reaction mixture after 60 minutes. Then similarly here, if we monitor the reaction mixture of the K4 methyltransferase, then only the reaction mixture after a long time actually will be approaching to the standard mixture that containing the proper product. Then using this kind of like the standard mixture as kind of like the index, we can see which would be the specific substrate of this enzyme. So we can help to discover like the particular substrate of a new enzyme, and by just simply monitoring the fluorescence signal change of a bunch of host gas pairs. Then the lysine methylation is the very straightforward target because it has the trimethylammonium head that is actually the intended gas for the cavitant. However, there were many different kinds of post-translational modifications, including phosphorylation. So this is actually a major type of PTM, and it actually regulated in response to physiological stimuli. So it's caused phosphorylation of proteins, and then this phosphorylation event could be translated to downstream proteins, like after the signal pathway is turned on. So this is like need to be strong, tightly regulated, and also it's actually the enzymes regulating protein phosphorylation are a large class of drug targets. In particular, the kinase. This enzyme is actually putting phosphate group on the proteins, actually forming a large class of drug targets. However, phosphate group, which is a negatively charged group, actually by itself, is not a good gas for deep cavitants. So then we can kind of think that whether it could work. In this case, we actually find at this point, we have found another type of gas, which is the phorogenic gas. It does not have a very common trimethylammonium head. However, it has a kind of similar structures. And this one also can bind to a cavitant. However, different than the fluorescence quenching we observed in the previous 2 cases, with this phorogenic molecule, once it binds to cavitant, its fluorescence will be turned on, and then the enhancement factor is actually pretty large. Then in this way, we also find that heavy metals actually can influence the fluorescence level of this gas and host and gas pair. So we're thinking it could be the heavy atom effect that quenching the fluorescence, or it could also be due to the cavitant that we used here. We call it TCC. Actually, it has the carboxyl group on the rim. So then this carboxyl group actually could chelate with the heavy metals, then actually would change probably the opening of the cavity, or it could be inserted in between the dye and then the phorogenic gas and the cavitant to modulate the fluorescence. In this case, by adding the additives, we think whether this type of metal cations as the additives could help us sense like the uncommon targets like a phosphorylated peptide. Then we think that there could be, in this case, then since the peptide, again, still could be, we chose to work on cationic peptide, just like the histone peptides, because the cationic peptide could interact with the cavitants electrostatically. Then in this case, then we have multiple interaction systems exist. So we could have the metal cavity interaction, peptide cavity interaction, and dye cavity interaction, or even a higher order of complex could exist. Then in this case, it's no longer just a simply indicate displacement for our fluorescence change. Rather, it's the competition between the fluorogenic dye and then the cationic peptide for binding to the cavitants. This is what we hypothesized, because we in general see like if the histone peptide is non-phosphorylated, the fluorescence when it mixed with the cavitin and DSMI, which is the fluorogenic gas, then the fluorescence always will decrease when it's lower than the phosphorylated peptide. So that's why we think that the phosphorylated peptide actually has a weaker interaction with the cavitin. Therefore, it actually has a less competition to the dye, to the fluorogenic gas to bind to cavitin. Therefore, they would see higher fluorescence. In this case, then when we have a phosphatase that remove the phosphate group from this histone peptide, then we should see over time, then we could see the fluorescence should decrease because there would be more non-phosphorylated peptide exist in the system. That means more competition between the peptide and then the dye to prevent the fluorogenic dye to bind to cavitin. Therefore, overall, the fluorescence will decrease. So then this again, single point would not work too well for complex mixture, right? So, and then we also now we have sensors that can sense like methylation. Then we also have sensors that can sense phosphorylation. So then it triggers us to think that we probably can use the sensor to study PTM crosstalk. That means the different types of post-translational modification exist on the peptide, but at a different location, and then they could actually affect a nearby PTM formation. For example, here what we demonstrate is that if the methylation on the K9 position, this number 9 lysine position, actually could affect the serum phosphorylation at the 10 position, at number 10 position on this peptide. So then we can see actually if it has the dimethylation on the lysine, the enzyme actually will not work so well on this peptide by putting the phosphorylation on it, then compared to the non-methylated peptide. So this is actually representing how the biological system could use like a nearby PTM to control the activity of the enzyme on putting on another type of PTM on the nearby amino acid. And then our sensor, since we have a specific sensor for methylation and also another sensor that senses the phosphorylation, we could actually tell the differences between methylation and phosphorylation. Then we could actually use our sensor to study the crosstalk of these different PTMs. But still, these are the cationic peptides. So it's again like although it's strictly not indicated displacement, it's the dye and target competition for the cavitin. Then we think that can this kind of, we call it indirect sensing strategy, be used to sense like electron-rich or negatively charged analytes? Then actually our work have proved that this is actually could work. So kind of like we have if we have in this case, it's again different than what we have seen before. So what we will show later is actually the dye molecule will interact with our target. Then also our cavitin can interact with this fluorescent gas or fluorogenic gas. Then this actually forming again another type of competition. So in the previous example, it's like the dye and target compete for cavitin binding, and here actually is it's a cavitin and target compete for interacting with the dye. So we screen like our different carcinogenic guests with several peptides, and we found out an interesting phenomenon that actually if the peptide, even though if it's an ionic one, if it has a relatively high hydrophobicity, and also if it has a tryptophan in the sequence, you could see enhanced fluorescence when the dye bind to the peptide. So we use molecular dynamics to simulate the binding between the dye and then this tryptophan containing peptide. And we found a certain structure actually have the dye molecule inserted between the arginine and then the tryptophan on the more relatively flexible peptide backbone. So, and then this kind of insertion probably that's the reason of why the fluorescence of this dye could be enhanced once it binds to the peptide. Then we think that we could probably take advantage of this fluorescence increase to sense peptide phosphorylation. So then we study the various interaction in our system. Like we can see actually the dye could bind to the peptide and causing fluorescence enhancement of the gas molecule. And then when we study the dye-peptide interaction, we could see the tryptophan emission actually could be affected by increasing the dye concentration. That also, again, indicating interaction between the dye and the peptide. So again, also the cavitand actually can kind of, here is showing the cavitand also could change the tryptophan emission and then indicating cavitand peptide binding. So using all these fluorescence curves, we can actually do curve fitting to obtain the empirical dissociation constant KD values. So we could see the binding actually is highly variable between the cavitand and the dye, the cavitand and peptide, as well as the dye with the peptide. Then once we add the cavitand and peptide to the dye solution, and then all this kind of fluorescence change, again, the profile will change. So in the, this possibly due to a formation of this triplex, like a tertiary complex, like we have the dye, peptide, and cavitand all bind together. Then that means we again can observe variable fluorescence on peptides if they have a single site phosphorylation, but then disphosphorylation at different location. So the 7 and 3 meaning is the number 7 and number 3 serine on this peptide sequence, or it could be a dimephosphorylated peptide. But only one site on this peptide is the activity site of the enzyme that we will study later. But anyway, we do not actually need a large array to differentiate all these different phosphorylation situations. So eventually we find out only 3 elements would be good enough to work on differentiating different phosphorylations. Then using, again, a different kind of supervised classification tool, we can see all this different differentiation, the differentiation of different phosphorylation peptides. And then if we use it to monitor the site-selective kinase activity, we can see this pKa only actually work on the sp7 side, but then not the sp3 side. So we can see following the time, we can see the from 0 hour to 48 hours, we can see this pK reaction mixture getting close to the sp7 peptide, the standard solution. And if we have the heat inactivated enzymes, actually we do not see any change of the fluorescence pattern of this reaction mixture. Then that actually meaning that the indirect sensing strategy can be applied to sense peptide phosphorylation, and then that does not need to be a cationic peptide anymore. Then it's actually, if the dye can interact with the target, we have another handle to design this kind of sensing array. Then actually this kind of indirect sensing strategy, before we apply to an ionic peptide, we actually have already used it to sense the different folding of short DNA. For example, for the G quadrupole structures. So this G4 structure, again, is another kind of function regulation of the nucleic acids in addition to its sequence. So then this kind of different folding could interact with proteins. So then the different structures can again implement gene regulation. Then for G4, it's very interesting that the G4 structure can have different orientation of the strands. So then that's called the different topology. So that a parallel, anti-parallel, and hybrid topology. And we have proved that using this different kind of host gas combinations, we actually can differentiate or just like a hybrid, in particular the parallel and hybrid as well as anti-parallel structures pretty well. So then here showing that like if we have 23 different G4 strands, we can classify this different folded topologies into 3 groups, and then only 1 out of the 18 samples in our data set actually was misclassified. Then using this kind of sensing array, we again can monitor actually the structure switch, like the topology switch in the G4 structure induced by changing of the cations. Like if we change the sodium, replace the sodium salt by potassium salt, we actually will change the anti-parallel structure to the hybrid structure for this particular G4 strand. Then we can monitor that using the interpreting the fluorescence pattern, and then we can differentiate the unfolded and then the G4 folding in a mixture, and then with increasing proportion of the G4, we could see our fluorescence pattern again change consistently. Using this concept, we can monitor the chemical modification on G4 structure, and we have achieved successful differentiation of many different folding types, like including like the imperfect G4 structure, hairpin structure, triplex, as well as the eye motif. However, for this example, what I want to introduce the concept of using like machine learning to improve differentiation. So when principal component analysis is like very straightforward and also unsupervised, so like produce a very true classification effect of our fluorescence patterns. When we have like a large number of dyes and host, as well as many different structures, then our data set becomes very complex. In that case, we actually have to rely on machine learning tools. So what we use here is called a support vector machine, and that can actually do recursive feature elimination. So through this SVM RFV approach, we actually can minimize a large array into a very effective and efficient array size, a small array size. And then we also can build the recognition, like classification model, so that when we have a known structure, we can put in this model to do structure folding prediction. So we have demonstrated that like not only just like an individual sensor may actually be used for sensing, like or we also can use an array of the host and guest to improve the differentiation power. And of course, if we can integrate with machine learning or artificial intelligence, I believe this could be even more powerful, for example, to overcome the complexity when we detect in biological media. And also we could enhance the selectivity to do more differentiation of smaller changes in biomolecules. So that's what my group has been continuing working on. So we want to rely on this kind of artificial AI tools. And also we would like to have more variety of host and gas interactions to see whether we could achieve even more diverse molecules, like detection of even more diverse molecules. So at the end, I would like to thank my groups, either those who worked in UCR already graduated, or the one currently working in my present group in USTC, and the support from both universities. Right now, I'm full-time USTC, but my previous career really benefited from UC Riverside. So I really appreciate that. And at the end, I want to take 1 minute to introduce the Precision Chemistry Journal that I'm the topical editor of. So this is a relatively new journal, and it's an open access. So if you submit your work by the end of this year, you can actually, we can waive the cost for open access publication. And many our journal actually focus on works that actually try to improve achieve higher precision in calculation, design of chemical synthesis, as well as analytical chemical measurements. So if you have relevant works, please consider our journal for submission. Currently, we actually have 4 topical issues, like special issues that calling for papers. So if your work falling in this kind of areas, please welcome to submit to our special issues. So at the end, thank you very much for your attention.